Now we are going to go over 5.1, 5.2, and 5.6 on page 189, and then 6.3 and 6.4 are on X-linked disorders on page 194. So starting with 5.1, the mother is IB little I, the father is IA little I. If you work a Punnett square using those genotypes, IB little I and IA little I, you're going to get all four of the genotypes that are listed here. Okay, this one looks funny. That B should be like a superscript, but it should be IAIB, IA little I, IB little I, and two little i's. And so it's a fourth chance that they will have a child that's any of the four blood types. Fourth chance that they'll have a child that's AB, A, B, or O. Okay, and then for 5.2, the genotype of the father is IAIA, and the genotype of the mother is little I. So since both the father and you don't even have to do a Punnett square for this one, because both the father and the mother only have one allele to give. The father can only give the child an IA. The mother can only give a little I. So all of their children are going to be heterozygous, IA little I, and they're going to have type A blood. So they're going to be heterozygous for type A blood. And the other one I want to say was 5.6. Yeah, a mother is type O, the father is type B. What are the potential blood types of their offspring? So what you have to do with the Punnett square for this is know that the mother's blood type is, or genotype is, two little i's, but the father can be IB or IB little I. So you actually have to work two Punnett square problems to answer this question. You have to work a Punnett square where the mother is two little i's and the father is IB. Then you also have to work one where the mother is two little i's and the father is IB little I. And the answer ends up being that the potential offspring can be blood type O or they can be blood type B. Now, for 6.3 and 6.4, which are sex-linked, so this 6.3 is about red-green color blindness, and it says the man is colorblind, the woman is a carrier. So the male or the father's genotype is X little cy, and the mother's genotype is X little c, and then they're just using an X with no, in class we did X big C, X little c. Well, this X with nothing is what we used. We used X big C for that. So this is basically saying the X little c is her chromosome that carries the colorblind trait, and the X with nothing, no notation there, it means for the normal, for the normal vision. So anyway, if you work a Punnett square with these two genotypes for the female and the male, the offspring can just be XY. You can see XY, which that X would be the normal gene because it doesn't have a C. Or you could have offspring that are X little cy, which means that they would be colorblind males. So this would be XY would be normal male, X little cy would be colorblind male. X little cx would be a carrier female, but she would have normal vision. And then 2X little c's would be a female that is colorblind. And there's a fourth chance of having all of these conditions. The problem for 6.4 is for another X-linked disorder called Duchenne muscular dystrophy. The male is XY. He does not have muscular dystrophy. And the female is a carrier. So that little d is the gene for muscular, or the allele for muscular dystrophy. So when you do a Punnett square, you can have two X's, which is a normal female who is not a carrier, You can have X little dx, which is a female who is normal, but she is a carrier for muscular dystrophy, and she can pass it on to her children. Then you have XY, which would be a normal male, or you could have a fourth chance of having X little dy, which is a male that does have Duchenne muscular dystrophy. Now, going back to the study guide, in lab 14, basically questions 1 through 5, let me check those out real quick. Questions 1 through 5 would be, I know these aren't numbered, but I remember them pretty much. This would be lab 14. Oh, sorry, I shouldn't be in that. All right, lab 14 is the one on DNA. And what we did in this lab was we isolated DNA from fruit or pumpkin. And then let me find that question. Here we go. Okay, so we isolated DNA from fruit, and we also did gel electrophoresis, and we looked at different DNA banding patterns and tried to, solve problems either with paternity cases or crime scenes. And another thing is you would need to know, what the components of DNA isolation that we use to isolate the DNA, like what is the detergent for, what is the alcohol for, what is the salt for. So we'll talk about that. Question number 1, what are the 3 parts that make up a nucleotide? And just to make sure you know, this was 1 through 5 on page 207 in the lab manual. So that goes along with this. The 3 parts that make up a nucleotide are a sugar, a phosphate, and a base. And the base can either be A, G, C, or U in RNA, and it can be A, G, C, or T in DNA. And that's not listed here, but they only have U listed, but for DNA it would be a T. All right, for number 2, the answer is E. For number 3, the purines are adenine and guanine. They have a double ring structure. The pyrimidines are cytosine and thymine, C and T. They have a single ring structure. Number 4, the first blank is transcription. Transcription is when DNA is copied into RNA, and that RNA is specifically messenger RNA. And then translation is in the second blank, and that is where the genetic code, the messenger RNA is used to code for an actual protein. Number 5, you pick one of the types of RNA, either mRNA, tRNA, or rRNA. mRNA stands for messenger RNA, tRNA stands for transfer RNA, and rRNA stands for ribosomal RNA. So the one that always attaches to specific amino acids, in other words, it transfers amino acids to the ribosome, that is tRNA. The nucleolus makes ribosomal RNA or rRNA. The one that works in triplet codes or triplets to form codons that code for specific amino acids, that would be the messenger RNA. A single strand of nucleotides whose bases are complementary to the DNA from which it was transcribed, that is messenger RNA. 2 subunits, that's rRNA, and then 3 bases that are complementary to the codon. We call them the anticodon. That would be tRNA. Okay, let's see what else. 1 through 5, and then be able to determine the complementary DNA strand if given the base sequence for the template strand. So if I give you a strand, for example, TAC, then it's going to be ATG. Just make sure you know A is complementary to T and C is complementary to G. And then if I give you the DNA, you should also be able to determine the messenger RNA sequence. In that case, you would do it just like when you are telling the complementary DNA strand. But since it's RNA, wherever you would have put a T, you put a U. Now, the rest of this says be able to determine the tRNA anticodon sequences. Just forget that. That's, we're not going to, in fact, I'm going to just take that off. I'm not going to take it off of what's posted on Moodle, but just for right now, you don't have to do that. Only be able to determine complementary DNA strand if I give you the template DNA, and be able to determine the messenger RNA sequence if I give you the template DNA. Then be able to use the codon chart to give me the amino acids. So just remember for the codon chart, and let's see if we've got a picture of it. Let's go back up here. I mean, it doesn't get easier than using a codon chart. I don't see a picture of it here, but basically, whatever your messenger RNA codon is, you just find it on the chart, and that's your amino acid. So you will have to do that. And the purpose of the detergent is to break down the phospholipid bilayer of the plasma membrane, to break down cell walls, to break down the nuclear envelope. So basically the plasma membrane, cell walls, and nuclear envelope. That's the purpose of the detergent. Detergent breaks down grease, and grease is a lipid. It's a fat or a lipid. So that detergent is going to help break down the cell walls, the plasma membrane, and the nuclear envelope. The salt is to separate the DNA from the histone proteins, because the chromosomes inside the nucleus are composed of DNA plus protein, so the salt will separate the DNA from the protein. And then the alcohol is used at the end to separate the DNA from solution based on density. If you remember, we poured the alcohol slowly down the side of our test tube, and what we ended up having was alcohol on top, DNA in the middle, and then our solution of water and fruit and detergent and salt, all of that, that we had filtered. That would have been on the bottom. So our DNA will separate from solution because it is actually less dense than alcohol, but more dense than water. I said that wrong. More dense than alcohol, but less dense than water. So it's going to be underneath the alcohol layer and on top of the water layer. So the alcohol was used to separate out the DNA based on density. Now, we've gone over this in class, but I'll just real quick go over it again with you. Make sure you can solve these problems. In the first problem, you have 4 different samples, but they're all the same individuals, the same mother, child, and alleged father. The only difference is you're seeing the banding pattern is different because even though it's the same DNA samples, it is different restriction enzymes that were used to cut those DNA samples. So what you're looking for is for each of the child's bands to match up either with the mother or the father. If you find one of the child's bands that doesn't have a match, it is not the father. So we were able to say with certainty that this alleged father is the father of the child. Okay, in the next problem, we were able to say that dad 1 was the father of twin 1 and dad 2 is the father of twin 2. We were also able to say that this father, and this is kind of cool because you can see the color picture. But we were able to say this father was not the biological father of the baby in the picture. In the sexual assault case, it was suspect 1 whose DNA matched up with the sperm DNA. And it was child 2 that was least likely to be the biological child of the couple. In this problem, the boy in question is the biological sibling of the brother and 2 sisters. Even though there's not a father's DNA, every one of the bands of the boy in question either match the mother or one of the siblings. For megabucks, it was Y's child, who is the biological child of IM megabucks. For the mix-up at the hospital, I always forget this one. Okay, so baby number, I always remember baby 2 is the Joneses, but baby number 1 is the Stevensons, and baby number 3 is the Smiths. All right, then we have, all right, in this situation, we have 2 biological children, a child that's only the mom's, Mrs. Hunch, and then a child that is adopted. Son 2 is adopted. Daughter 1 and son 1 are biological offspring of both parents. And then daughter 2 is only Mrs. Hunch's. It's really helpful because the dad's bands are not shaded in. So it's easy to do this problem. In the challenge question, it's asking you to determine which lane represents the DNA fingerprint of the father, and the other 4 would be the father's children. So it's lane 3. Lane 3 represents the DNA fingerprint of the father. Then we have some karyotypes I'm just going to go over with you. This karyotype shows that the child is male because the pair number 23 is an X and a Y instead of 2 X's. And this child has Down syndrome because it has 3 chromosomes instead of 2 at chromosome pair number 21. So it's called trisomy 21 or Down syndrome. The next one is okay, so this is a male because the presence of a Y determines that it's a male. And it has XXY, which means this is a condition called Klinefelter syndrome. If it was XYY, it would be a condition called Jacob syndrome. But it's 2 X's and a Y, which is Klinefelter syndrome. All right, this one is just 1 X, so this is a female, but she is missing a chromosome at pair 23, so she has Turner syndrome. Turner syndrome. And I don't think I'm going to ask you about the karyotypes because we didn't really do that much in class, but just in case. So that is the Lab Practical 2 review. Make sure you let me know if you have any questions about any of those.